ये आवाज नहीं आ रही, जरा देख लो। चलो ठीक।
Hello, ma'am. Am I audible now?
Yes, Pradyuman, please continue.
Okay, ma'am. This is to inform you all that the ceremony is being recorded. I request all the participants to keep their microphone muted and video turned off throughout the event. Henry Ford has said, anyone who stops learning is old, whether 20 or 80. Anyone who keeps learning stays young. The greatest thing in life is to keep your mind young. I, Pradyum, on behalf of Department of Zoology, extend my warm welcome to all the dignitaries, faculty, and participants to the inauguration ceremony of the online short-term certificate course, Immunological Techniques and Their Applications, organized by Department of Zoology of Bhaskaracharya College of Applied Sciences in collaboration with Department of Zoology, University of Delhi, under the aegis of IQAC. This course is also an initiative under Vidya Vistar scheme of University of Delhi for spreading the knowledge with faculty and students of Department of Zoology, Indira Gandhi Government College, Tezu, Arunachal Pradesh. I would now like to request Professor Anil Kumar Bali, teacher in charge, Department of Zoology, to address the audience. Sir.
Professor Satyajit Rath, eminent immunologist, Indian Institute of Science, Education and Research, Pune. Professor Balram Pani, principal of our college and dean of colleges, University of Delhi. Faculty and student of Indira Gandhi Government College, Tezu, Arunachal Pradesh, under Vidya Vistar scheme of the University of Delhi, resource persons, my colleagues, participants, and dear students. I welcome you all on the occasion of inaugural function of online short-term course on immunological techniques and their applications. Professor Rath will deliver the inaugural talk on the promise and problem of immunology. Professor Rath is a medical doctor who entered into research and has number of years of postdoctoral research experience in the field of immunology in various laboratories abroad. He has 32 publications in the international journals. He is also associated with National Institute of Immunology as adjunct faculty. Immunology is an emerging field which has also become the part of molecular biology in elucidating gene and its function. For example, immunological screening uses specific antibodies to detect expressed gene products. All the participants shall have the opportunity to learn the various aspects of immunology. I welcome you once again. Thank you.
I extend my sincere thanks to you, sir. I would welcome the best wishes of our principal, sir, Professor Balaram Pani, who is also Dean of Colleges and Chairman, Governing Body of School of Open Learning, University of Delhi, who has sent his best wishes as he could not be present here due to his tight schedule. I now welcome Dr. Chatwan Tesya, Head of Department of Zoology, Indira Gandhi Government College, Tezu, Arunachal Pradesh, to address our participants. Ma'am.
Hello, everyone. I think I'm just audible to all of you.
Yes, ma'am, you're audible.
Okay? Thank you so much. A very good evening, everyone. So, sending good wishes all the way from Indira Gandhi Government College, Arunachal Pradesh. First of all, I would like to offer my sincerest thanks to the organizing team of this program, that is short-term certificate course on immunological techniques and their applications. And also, I would like to thank the convener of this program, Dr. Anil Bali sir, as well as Rituparna Basak madam, because it's because of their coordination that we are a part of this program. And I, on behalf of the faculty members of Department of Zoology, as well as the students who are participants out here, for the invite which you have sent us. And I would like to take this opportunity again and this platform for thanking University of Delhi for the scheme, that is Vidya Vistar scheme, because of which we are a part of this program. Regarding this course, we are so much looking forward to it. And I just hope that by the end of this 40 hours session, we'll be kind of unreached with it. Because we know that when we just read some bookish, syllabus, when you just go through your syllabus and all this, it's like there are some techniques about which we know, and there are much more about which we have never heard about. So this will be a really good opportunity for the faculties, for us, as well as the students. They'll be benefited a lot. So once again, I would like to thank all of you back there in Bhaskar Acharya College of Applied Sciences for this opportunity. Thank you so much, everyone.
Thank you so much, ma'am. It was a great experience listening to you. Now I would like to welcome Dr. Rituparna Basag, Assistant Professor, Department of Zoology, Bhaskaracharya College of Applied Sciences, the coordinator of this short-term course, to acquaint us with the scope of this course. Ma'am.
Thank you, Pradyuman. Good evening, everyone. I welcome all dignitaries, resource persons, principals, sir, faculty members of Bhaskaracharya, and the participants to the inaugural ceremony of the online short-term certificate course, Immunological Techniques and Their Applications, that is being organized by our college in collaboration with Department of Zoology, University of Delhi. The course is also an initiative under Vidya Vistar scheme of University of Delhi, through which our department wishes to share the knowledge and expertise with faculty and students of Indira Gandhi Government College of Tezu. In the present world, the pandemic caused by coronavirus has quite significantly showed us the importance of medical diagnostics and the development of drugs and vaccines. The whole research community has felt the importance that a very productive research is required in the field of immunology. Most of the diagnostic techniques that we have today are based on immunology. We know all of their names, but we hardly know their basics. Hence, the present online short-term course, it aims to impart in-depth knowledge, the theoretical knowledge of these techniques, ranging from very simple techniques such as development of polyclonal and monoclonal antibodies, immunodiffusion, ELISA, etc., to the advanced ones such as confocal microscopy, chromatin immunoprecipitation, FACS, etc. The course will also focus on the detailed procedure of the techniques so that the participants may gain a bit of technical experience, which is not equivalent to hands-on experience, but in the current scenario, this is the best that we can do. The course has received till now registration of 81 participants that includes teachers and faculties from various universities throughout the country. The total duration of the course is 40 hours, and it starts with today's inaugural session when our eminent immunologist, Dr. Satyajit Rath, will be the invited speaker. Sir, thank you for accepting our invitation and honoring us with your presence.
Thank you.
Thank you, sir. There are 10 lecture sessions that will be conducted only on Saturdays and Sundays from tomorrow till November 7th. The lectures are being conducted by some of the most experienced professors, scientists, and research scholars of Department of Zoology, University of Delhi, and National Institute of Immunology, Delhi. I am very thankful to Professor Madan Mohan Chaturvedi, Professor Anju Srivastava, and the research scholars of Department of Zoology, University of Delhi, and also to Dr. Tanmay Majumdar from National Institute of Immunology for accepting our invite. The course will end with valedictory session on November 8, wherein participants who have fully completed the course will be awarded with participation certificate. The organizing committee has also decided to award top 5 participants with merit certificate based on their assessment marks, which will also be one of the component of this course. I sincerely hope that the course will be very beneficial to the students, not only in increasing their knowledge, but also to create an interest for research. I extend my best wishes to all the participants, resource persons, and the organizing team. Thank you. Over to you, Pradeep.
Hello, ma'am. Am I audible?
Yes, Pradeep.
Yeah. Thank you so much, ma'am. It was a great experience listening to you. I would now take the opportunity of welcoming our respected invited speaker, Dr. Satyajit Rath, an asset to our nation, who is currently associated with Indian Institute of Science Education and Research, Pune. As an avid researcher, he has the experience of practicing at Haskins Institute, Mumbai, India, London School of Hygiene and Tropical Medicine, London, UK, Brandeis University, Waltham, USA, and Yale University. And activation of T, B, and antigen presenting cells, APCs, of the myelinoid lineage using a variety of interlinked experimental systems and approaches. He is affiliated to National Institute of Immunology and University of Mumbai, apart from his loco opus and several other places. Once again, I welcome Dr. Satyajit Rath. Sir.
Thank you very much. I extend my best wishes to all the participants of the workshop and my congratulations to the organizers and the resource persons for the efforts that they are putting in under difficult circumstances. Now, let me point out that unlike all the other speakers, I'm supposed to simply do an inauguration ceremony invited lecture. So I'm not going to provide any specific immunological technique. And therefore, I thought what I would do in a conversation in half an hour is to talk a little more generally about the promise and the problems of immunotechnologies, which is all said and done what you're talking about over the next 3 weekends, as Dr. Prashant pointed out. So every time one thinks about this phrase of immunological techniques, there are 2 ways I suggest to think about what is an immunological technique. So one way of thinking about it is any technique that is used in an immunological investigation, meaning any technique that is used in an investigation to work out immune mechanisms and immune responses is an immunological technique. And that really doesn't work. It doesn't work because a lot of those techniques are general techniques in biochemistry, in molecular biology, in cell biology, and so on and so forth. So, by and large, when we think about immunotechnologies or immunological techniques, what we really mean is technologies that involve the use of immunological reagents, immunological tools. So the question then becomes, what is an immunological tool? And the straightforward answer to that, of course, is antibodies. So by and large, what you are really going to talk about is antibodies. The next 3 weekends, with the exception of 1 day, is going to be about antibodies. That 1 day of exception we should take note of. Because that one day of exception, I think Dr. Olivia Roy will anchor on the 7th of November, is about phagocytosis. So let's go back a century and a quarter to the beginnings of analysis of immune responses. And there were 2 Nobel Prizes in fairly quick succession in the early years of the 20th century. One was about phagocytes and the process of phagocytosis, and the other was about antibodies and clinical use of antibodies. And you can go and, I'm not going to use the word Google, you can go and do net searching for all of that. But the reality, therefore, is that both of those we have known about for a long period of time. The difference between phagocytosis as a biological property, a biological phenomenon, and methods for assessing phagocytosis versus antibodies is quite striking. Phagocytosis is a self-contained biological process. So the only time you use the technique of phagocytosis. I'm sorry, was there a question? So the technique of phagocytosis is going to be useful only in the context of measuring phagocytotic ability of cells of the immune system, neutrophils by and large, but macrophages and to a far lesser extent dendritic cells also. But the reality is that the use of the phagocytotic assay method is restricted. So what's so special about antibodies that we tend to use antibodies in almost half the methods in a clinical biochemistry laboratory? So in order to begin to understand that, we're going to go from the early 1900s to the mid 1900s, from about when somebody as prehistorically old as me was born in the 50s, by which time 2 researchers, one a physician in New York named Solomon Burson, and the other a woman, and please all women students, faculty take note, a woman, unique faculty member at the University of Illinois, Urbana-Champaign in the United States of America, had begun to work together. What were they working on? They were not working on immunology. They were working on endocrinology, actually clinical endocrinology, hormones and hormone biology. What they really wanted to do was to measure hormone levels. Now, how do you measure hormone levels, particularly peptide hormone levels, insulin? How do you measure hormone levels? So the straightforward method for them was to take a bioactivity assay, to take primary cell cultures, tumor cell cultures, to put the hormone in and to show that a certain activity that was measurable could be triggered by the hormone, to titrate the input, to build a standard curve, and therefore to measure against a standard curve built by a known titration of a standard preparation of the hormone in a bioactivity assay to build the concentration, to read the concentration in an unknown sample, in a test sample. Here was the problem. Nobody was quite sure that the bioactivity in a complex biological fluid, in blood, for example, in serum, insulin levels in serum, were not being modified by other components of the serum. How do you know that? So how do you know that the concentration of insulin in a test serum sample was the only thing that was determining the bioactivity of insulin in that sample, or other hormones, any other hormones you like to name? So these 2 researchers, Solomon Burson and Rosalind Yalow, were working together and talking to each other about this, and they said, can we measure insulin without invoking its biological activity? In other words, is there a probe for insulin that has nothing to do with its biological activity? Now, its biological activity is through its receptor. How are the insulin and insulin receptors specific to each other? Because over evolutionary time, they fit each other in specific ways. In other words, they bind each other with great biochemical structural specificity. Fine. Can we develop, independent of the cell, independent of the receptor, a surrogate receptor for insulin that's soluble, that's aqueous phase, that we can develop and that we can use to bind insulin and to pull it down in a precipitate and therefore to measure insulin concentrations? And the short answer to that was no, because we don't know what the receptor is. Even when we know what the receptor is, this is the 50s. Remember, even when we know what the receptor is, nobody can generate cell-free receptors because nobody's invented recombinant DNA technologies as yet. We just haven't gotten that far. Watson and Crick are still running around from one pub to another in Cambridge. How do you do this? And then suddenly, Emil von Behring, Paul Ehrlich, Karl-Heinz Steiner, and all the work of 50 years earlier became useful. Why did it become useful? Because what they had pointed out was that if you put a biological macromolecule under appropriate circumstances into an animal, the animal made proteins that specifically bound to this biological macromolecule. And if that happened, that's effectively, as far as biochemistry of binding is concerned, that's effectively a surrogate receptor. That's how Rosalind Yalow and Solomon Burson thought about antibodies. So how they used antibodies was as surrogate receptors. They made antibodies against insulin. They used radio-labeled insulin. In fact, Rosalind Yalow started out life as a radiochemist, as a radioisotope physicist. And she tagged the insulin with a radioisotope, used antibodies, and showed that when antibodies were precipitated using standard biochemical methodologies of salt-based precipitation, that radio-labeled insulin came down in a standard curve. And now, in straightforward biochemistry, when unlabeled insulin was added to this mix, it inhibited the binding of radio-labeled insulin. So what they built was an inhibition radioimmunoassay to measure concentrations of insulin. And hey, did it work? Yeah, it worked superbly well. Nice, right? So that's Yalow and Burson. And tomorrow you will talk to begin with about monoclonal and polyclonal antibodies. What Yalow and Burson were using were polyclonal antibodies. Solomon Burson died in the early 70s, 4 or 5 years later in 1977. Rosalind Yalow got a Nobel Prize for physiology or medicine for the radioimmunoassay. Because it was very clear from their work that the antibodies that immunologists had been messing around with for 50 years had uses that were far broader than simply trying to figure out how the immune system protects us against infectious diseases. Antibodies could be used as structurally specific tools and probes for a variety of biological macromolecules. So what were the promises of these antibodies? They were easy to make because all you had to do was to take the target, insulin, for example, inject it under appropriate circumstances, meaning in an adjuvant. In this COVID era, everybody is familiar with what an adjuvant is, at least notionally. Inject it into animals in an adjuvant. The animals generated antibodies. You could purify the antibodies. These were polyclonal antibodies, and you could use these polyclonal antibodies, and you could build immunoassays. You could build radioimmunoassays if you used radioisotopes as a probe. If instead of attaching radioisotopes either to target or to antibody, I don't tend to use the word antigen because it is much misunderstood, simply the target of antibodies. So you could attach the label, the detection label, to the target or to the antibody. The detection label could be a radioisotope or an enzyme, for example, or a fluorescent probe in subsequent years. You could attach it to the target or to the antibody. You could use this combination either to detect material in your test sample directly by, for example, coating plates with antibody, putting either standard target or test sample onto this, washing all the rest of it off, using labeled antibody and detecting it in a sandwich assay, which is not at all what Rosalind Yalow was doing in her original radioimmunoassays. You could build all sorts of detection systems that have gotten fancier and fancier and fancier and fancier over the subsequent decades. But the core is you could let the random complementary receptor generation capacity of the immune system in an animal to make specific probes for you. Great. So you could generate any specificity. We had the first half of the 20th century had shown that you could take unnatural molecules that have never existed on Earth. Chemists could build those, and once they were built, you could make them into immunogens, inject them into animals, and the animals would make antibodies against those. These recognitions by antibodies were precise almost as receptors, and were very high affinity. And because you could label either target or antibody, you could now build layers of amplification. So let me give you an example. I put an antibody on a solid phase support. Then I put target. To this target, I add a second antibody. But in that second antibody, which binds to bound target, I label it not with a radioisotope, not with an enzyme or perhaps with enzyme, but with something like, say, biotin. And then I put a biotin binding reagent, streptavidin, onto this. What I'm doing is amplifying, because to one target, one antibody binds, right? To that antibody, depending on how many biotin residues are bound to one antibody protein molecule, that many more streptavidin molecules bind. I can now put anti-streptavidin antibodies onto that, and I can therefore go on amplifying the signal. Right? This was fabulous. This came because of the radioimmunoassay. So the techniques of immunodiffusion and immunoprecipitation that people were using in the first half of the 20th century hadn't really thought about the major technological possibilities inherent in this amplification, in this layer by layer amplification. And that amplification has come to be a major feature of antibody-based immunotechnologies. All of this was beautiful. But as I pointed out, I will talk about, there were not just promises, there were problems. So the first problem was, what we are interested in detecting is biologically active molecules. I keep going back to Rosalind Yalow, the biologically active molecule of insulin. You want to measure bioactive insulin, but the antibody-based detection does not guarantee you bioactive insulin. Because why? Because protein folding is a huge problem in structural biology. You don't always have a protein that is completely properly folded or completely non-folded. You can have in-between states, you can have alternate states. And as a result, what you end up having is situations where the protein may not be biologically active, but may still be detectable by antibodies. And this gap is something that we still worry about. So let me point out how we worry about it in the current context. In COVID-19, in the pandemic, everybody's heard of the rapid antigen test. The rapid antigen test detects viral protein using antibodies. But the presence of a viral protein does not assure the presence of infectious virus. You may detect viral protein, but that's not the same thing as detecting infectious virus. That gap becomes a problem in the interpretation of the tests and the assays that we are designing. So that's one problem. A second problem is that I'm making antibodies. I have two horses, for example. I put human insulin into horses. The two horses make anti-insulin antibodies. But because they are making anti-insulin antibodies through a process that involves random mutagenesis, the antibody pools that they make are not identical to each other. In fact, we know that they are non-identical to each other, which creates a manufacturing problem. Because if I'm going to make a kit by immunizing horses, every new horse I immunize is making a slightly different reagent for me that will need to be re-optimized, retested, re-standardized. This is a nightmare. Every time we use a polyclonal antibody, we run into these difficulties. And what you will hear from your resource people, especially those involved in chromatin immunoprecipitation assays and in related assays where still a lot of polyclonal antibody usage takes place, is that some reagents, even from renowned commercial companies and resources, work better than others. In fact, a lot of retractions of immune methodology-based papers from literature are based on the fact that, oh, we thought that the reagent was precipitating, but we haven't been able to reproduce it because, you know, we had a batch of antibody that worked well, but now we have batches that don't work as well. So that becomes a huge problem. And that problem was something that was resolved. Remember, 1977, Rosalind Yallow's Nobel Prize for radioimmunoassays. But by that time, this problem of polyclonal antibodies and batch-to-batch variation, not just variation in the antibody profiles, but in the antibody binding affinities or average avidities, was solved, and was solved from a completely different direction altogether. It was solved in Cambridge by Jorzis Kohler and Cesar Melstein in a celebrated paper in Nature 1974 or 75. And about 10 years later, they got the Nobel Prize in 1984 for making monoclonal antibodies, or for what everybody refers to as the hybridoma technology. And what that led to was you could take one specific B cell antibody, one specific B cell receptor that could be genetically sequenced and identified, that could be put into a cell line, and from that cell line, stably, week after week, month after month, year after year, you could make the same protein monoclonal antibody. And this marked a shift in manufacturing and reagent stability terms. It created reproducibility, but it had a cost. I keep pointing out that there are promises and there are problems. So it had a cost. What was the cost? The cost was, if I take a large molecule, right, a large protein against which I want to make antibodies, chicken egg albumin. Why one would want to make antibodies against chicken egg albumin? Only immunologists know, nobody else knows. Who's interested? But anyway, chicken egg albumin. Now, chicken egg albumin is a folded globular protein, and an antibody can bind to many specific shapes on the surface of the chicken egg albumin molecule. Therefore, a polyclonal antibody mixture is a mixture that binds to all sorts of these surface regions. Let us take a relevant target. Let us take SARS-CoV-2 spike protein. And I can take SARS-CoV-2 spike protein. People across the world have done that, immunize animals with it, and animals make polyclonal antibodies. And those polyclonal antibodies bind to a number of different molecular regions of the spike protein of SARS-CoV-2. As a result, because you have this great diversity of binding, if there is a mutation in 1, 2, 3, 4 locations in the SARS-CoV-2 spike protein, as we know there have been, it does reduce the binding ability and efficiency of the polyclonal antibody a bit, but you still get pretty good binding because most antibodies will continue to bind. But if I take a monoclonal antibody, then that entire antibody preparation consists of completely identical immunoglobulin protein molecules, all of which bind to only one and only one location on the viral spike protein. As a result, do you get beautiful binding? Yes. Do you get very cleanly detectable binding? Yes. Do you get very low background signal noise? Yes. But if there is a mutation at the location at which your monoclonal antibody binds, your monoclonal antibody can suddenly lose reactivity. That means that small changes in target create a huge problem in detection. So as you can see, this almost excessive specificity becomes a problem. And therefore, what I strongly urge all of you to think about as you go through the 3 weekends of the course is how can you leverage the strengths of the polyclonal antibody approach, the strengths of the monoclonal antibody approach, and yet compensate for the limitations of both approaches. It's worth thinking about. It's the kind of question that immunotechnologies can use innovative thinking in. So that's why I'm urging you to think about that. Let me point out another issue that really a problem that immunologists tend to hide. And that problem is that when you use quantification, when you say, I have built a sandwich ELISA, which is a very common immunological technique for a viral protein, and I'm detecting viral protein I have a standard curve, and using that standard curve, I'm detecting the viral protein in a test sample, and I can tell you what the concentration of the viral protein in that test sample is. And in reality, the very strength and limitation landscape of antibodies as tools makes it almost impossible to rely on the absolute measurement capacity of immunological techniques. What you're doing is getting relative measurements. In other words, you're not getting a micrograms per ml concentration. What you're getting is sort of microgram equivalents per ml of approximate concentration. This is because of variable affinities. It's because of variable slopes of binding curves. In an inaugural session, I really shouldn't go into more detail than that, but I would urge you to ask your resource people over the next 2.5 weekends that you'll be talking about antibody-based assays about these sorts of limitations and the methodologies to at least compensate for these limitations as you begin thinking about immunological assays. So that's my sort of second set of problems about the immunotechnologies using antibodies. Finally, let me introduce to you a kind of immunological reagent and an immunotechnology that is likely to be more and more interesting in the clinical laboratory of the future, although it is currently not available at all. And in fact, the 3 weekends that you're going to be dealing with them don't even mention that tool, leave alone describe it in any shape or form. And in order to begin to introduce you to that notion, simply to introduce you to that notion, let me remind everybody that antibodies are made by one arm of the adaptive immune response, right? And this is not about expecting any of you to know immunology, but simply to know that the adaptive immune system has B cells and T cells. And both B cells and T cells have exquisitely specific target recognition capability. And secreted forms of the B cell receptor that B cells use for recognizing target are antibodies, which is what we are using. So what do T cells recognize? And why is it that we never hear of secreted forms of the T cell receptor in immunological technologies, in immunological methods? And that's a question that you might want to ask the immunologists who are serving as resource people. Rather than beginning to answer that question directly, let me point out where T cell receptor based detection systems would be useful. There are 2 places where they would be useful. Number one, it's becoming more and more evident that autoimmune diseases are driven not simply by autoantibodies, but also by autoreactive T cells. And therefore, we have a situation in which we need to be able to detect the formation of autoreactive T cell targets, meaning the targets that T cells recognize. And therefore, the nicest way of detecting those, you'd think, are T cell receptors, recombinant soluble T cell receptors functioning exactly like antibodies in the clinical laboratory. These are not used, they haven't been developed, they are unlikely to be developed in the near future. And as I said, you might want to talk to your resource people about exactly why T cell receptors don't work as probes and tools anywhere near as well as B cell receptors, antibodies, work as probes and tools. But here is the other end of that recognition dyad. Remember, that recognition is T cells recognizing target. Target is on cells of the body. Cells that are virus infected, such as those infected with SARS-CoV-2 virus, or cells that are being damaged as targets of autoreactive inflammation. In either case, cells are being recognized by specific T cells. So, can we identify T cells that recognize particular targets? We measure antibodies in circulation in vaccinated individuals. If you've had Covishield or Covaxin, you can go to the laboratory and for completely no good reason whatsoever, except for anxiety, get your blood tested to ask if you are now seropositive for antibody. How is that antibody tested? It's tested by an equivalent of Rosalind Yallow and Solomon Burson's radioimmunoassay, an ELISA of sorts. Please don't go get it done because it doesn't really tell you anything terribly useful other than increasing your anxiety. But the reality is that you can get it tested. So we know that you have a B cell response to the vaccine. How does one test if you have a T cell response to the vaccine or not? What do T cells recognize on the surface of virus infected or autoimmune target cells? And what T cells recognize is not just viral peptides, but viral peptides bound to HLA molecules present on the cell surface. So what you have is a peptide HLA complex. And what is emerging as a technology, as an immunological technique of the future, which will become more and more obvious in the clinical diagnostic laboratory, is building surrogates of peptide HLA complexes. They are usually built as multimers, as they were being sold as tetramers, but now they're being simply tested as trimer, tetramer, heptamer, etc., etc. So let's just call them multimers. As peptide HLA multimers used as immunological probes to detect either virus-specific or bacterial-specific or autoreactive T cells in the body, to identify the T cells, to identify their numbers, to identify their frequencies, to identify their functionalities, and to use that information in the context of a clinical or an epidemiological situation to make predictions about susceptibility or resistance to either infectious or autoimmune disease. And that technology is going to be an immunotechnology of the future that at least some of you, no matter where you land up, whether you land up in a diagnostic laboratory, whether you land up in a biotech company, whether you land up in a regulatory agency, whether you land up in a teaching institution, will become more and more familiar with. In fact, you will become much more familiar with than old people like me would be. So that's the range of immunotechnologies, their promise and their problems, that I wanted to remind everybody of as a prelude to this fascinating 3-weekend long workshop that you have on immunological methods and their techniques and their applications. I hope this is useful as context and background, and I'd be happy to answer questions if there are any. I will, since Dr. Poshak had said about a half an hour, I've already exceeded that. So let me stop here and thank all of you for your attention.
I extend my sincere gratitude towards you, sir. It was undoubtedly an informative and thought-provoking session, and I'm sure our audience have so much to take with them today. Thank you, sir. I would now like to request the participants to raise their hand if they have anything to ask. Attendees will be requested to unmute and ask their queries one by one accordingly. Yes, ma'am.
Thank you so much, sir, for coming to this inaugural ceremony. Sir, I really realize this fact that we have not included any component of the T cell system. Even we tried to include innate immunity, but we could not include this part. Thank you for giving us that preview. Sir, I have a very simple question, which while teaching, I often come to the query with myself only. Sir, can you just tell me how come autoreactive T cells are released when they have all the scrutiny system in thymus? Why do they escape it? So your mic is mute.
So let me respond to both. I brought up the T cell component because I think that for many of you in the audience, it is going to be a technology of the future that, as I said, old people like me really don't know well enough. On the other hand, as Dr. Poshak pointed out, there is already an innate receptor-based immune molecule technology that has absolutely fascinating implications outside immunology, and that has been used in the clinical diagnostic laboratory for a very, very long time, and that's complement measurements, which is part of the innate immune measurements that Poshak was talking about. And for all of you as participants, it's worth thinking about how we detect complement versus how we detect antibodies, and how complement detection can be amplified just as antibody detection can be amplified. And let me just make a teaching statement that antibody detection amplification is by and large non-enzymatic. Complement detection amplification is in common with polymerase chain reaction-based technologies for nucleic acid sequence detection, enzymatic in its amplification step. And these are similarities and differences that you might want to think about in a methodological workshop. That said, the question of autoreactivity. So in the first place, it's a fallacy to think that none of us as healthy people have any autoreactive B cells or T cells circulating. All of us have autoreactive B cells and T cells circulating, for the simple reason that no biological filter, no biological mechanism is 100% accurate. There is no such thing. Remember, SARS-CoV-2 as a virus, it's a coronavirus, and therefore, even though it's an RNA virus, it has some proofreading capacity, which means that it's not as inaccurate as, say, HIV or influenza viruses, but it still has an error rate. There is no biological enzyme that does not have off-target effects. Similarly, at the cellular level, there is no cellular mechanism in the body that is 100% going to filter out every last autoreactive T cell. That's just not going to happen. There is no negative selection system that's going to remove every last autoreactive B cell. So the question is, are there multiple mutually redundant pathways in the development of B cells and T cells that allow, by and large, autoreactivity to be kept under check? And the answer is yes, there are. So there are a large number of systems. In the first place, there is straightforward clonal removal. Autoreactive B and T cells are killed off. In the second place, there is clonal anergy. Forget about receptor editing, but there is clonal anergy, which actually turns them off but doesn't kill them. Now, this is both useful and problematic. It is useful because they can generate antibody responses against, for example, viral targets. This has been shown. But it's also problematic because, hey, what if they begin to respond to the autoreactive target? And they, in fact, sometimes do begin to respond to autoreactive targets. Exactly similarly, we have suppressor mechanisms, such as regulatory T cells, that suppress both autoreactive B cell and autoreactive T cell responses. But what if suppressor T cells, Tregs, regulatory T cells, don't just lose a little bit of quantitative function and you'll start getting autoreactivity? So there are a whole range of ways in which we can start seeing autoreactivity. All autoreactivity does not necessarily lead to disease. So let me give an example. For the bulk of our audience today, which is young and healthy, versus me, clearly not young and clearly not as healthy as them, if we take you and me, we all take exactly the same shot of tetanus vaccine. But what we measure in response is both anti-tetanus antibodies and autoreactive antibodies, rheumatoid factor antibody levels. Over succeeding weeks, both you and I will have an increase in the levels of tetanus toxin specific antibodies responding to the vaccine. I will respond a little bit less well. You will, in all likelihood, respond a little bit better. But you will respond very, very poorly in terms of rheumatoid factor, in terms of autoreactive antibodies, whereas me, in all likelihood, my levels of rheumatoid factor will go up in response to a tetanus toxoid vaccine. Why that happens is going to take much more than this conversation to explain. But the point I'm trying to make is there is a difference in the quantitative efficiency of regulation of autoreactivity between young people and elderly people. And yet, that does not mean that I have an autoimmune disease. All of these are quantitative shifts rather than qualitative yes-no responses, and the nuances of mechanisms, therefore, will need to be kept in mind in quantitative terms.
Thank you, sir.
Thank you, sir. We have one more attendee who wants to ask a question. It's Harsh Panwar. Harsh.
Good afternoon, sir. So when we start testing a vaccine or developing a vaccine, we first test it on animals and then it is rolled out to humans. But sir, then why do we need a separate vaccine for animals? Like recently, Russia is starting testing that Carnico vaccine on animals.
So it's a good question. And the difficulty, I think, is a little bit to do with terminology. We say we test in animals and then test in humans. But really, all we are doing is we are testing in one animal and then we are testing in another animal, right? Like you and me. In other words, Homo sapiens is just another animal. The fact of the matter is that when we test vaccines, what we are testing is three separate components. One, we are testing safety, correct? So, second, we are testing immunogenicity. For most vaccines, such as the COVID vaccines, we're testing whether antibody levels are being generated or not. And third, we are testing protective ability. Whether antibodies are generated or not, does vaccination protect against actual disease? Now, since we are trying to make a vaccine for a human disease, what are we doing? We develop a vaccine, we test it in mice for safety. Fine, they're safe. We still need safety tested in humans because there may be differences between mice and humans. So we do a phase 1 safety testing in humans. Number two, we've shown in mice, in guinea pigs, common laboratory animals which are available and accessible, that they make antibodies, good levels of antibodies. We do a phase 2 testing in humans, again, to confirm that humans make antibodies. But the phase 3, we cannot do in mice because SARS-CoV-2, the COVID-19 virus, does not infect mice. Even if we sort of, with much genetic manipulative contortion, build mice in which there can be infection, that's not real infection and real disease. So we end up needing to do testing for protective efficacy, so-called phase 3 trials, in that animal species in which the disease naturally happens and in which you're trying to induce protection. Correct? Now, by and large, when you are trying to build a vaccine in either humans or in livestock animals, in poultry, in pigs and buffaloes and goats, in sheep, in cows, in mink, in whatever, none of those animals are common laboratory animals. So what you do in them is parallel to what you do in humans for making a human vaccine. Empirical testing is always going to be needed because there are always going to be unexpected outcomes. So this isn't about have you tested in animals? It's about which animals you've tested in and does the disease naturally occur in that animal species, and is that animal species the species you're trying to protect against? If it's not, and it commonly isn't, then phase 1 and phase 2 are always going to be done both in an animal model in the laboratory and in your target animal, whether it is human or non-human livestock, and phase 3 is always going to be tested in the actual target animal. I hope that addresses the question at least a little bit.
Yes, sir. Thank you, sir. I have one more question. So why do we divide when testing people in
In all fairness, can we take somebody else's and then come back to you?
Yes, sir. Sure.
Go ahead. Pallavi Gupta.
Thank you, sir. So my question is that can we use antibodies which are generated in animals? So if we use them in any human treatment for any kind of shot, so if yes, then it won't be act as an antigen for human.
So the answer to both, it's a great question. It's what people did for rabies. Remember, it's what people did with antiserum, horse antiserum. When I was in medical college, where I'm sitting in Pune, I mean, I spent 27 years at the National Institute of Immunology, but where I have been for the past 2-3 years in Pune, there were horse farms here in which people were injecting diphtheria toxin, tetanus toxin, botulinum toxin, and generating antisera for use in people with disease for injection. So does animal-generated antiserum work in human disease? Yes, it did. It does. Monoclonal antibodies work to a certain extent, not terribly well, but to a certain extent. Incidentally, please, everybody, convalescent plasma therapy and monoclonal antibodies do not work in severely ill COVID-19 people. That's simply a matter of information. Again, why that is so will take much too long to discuss here, but it doesn't. Trials show that it doesn't. But that said, you're perfectly right. It's been used like that. It's been used like that for a century. And the answer to your second question is also yes. That's why if you had one dose of a horse serum, you were at great risk if you needed to be given another dose of the horse serum because you got massive allergic response because you made antibodies against horse antibodies. And they generated massive allergic responses. This is why humanizing antibodies, particularly humanizing monoclonal antibodies, so that you can now produce them in cell lines in established fermenter technologies, you can create large amounts of them, industrial levels of them, and yet they are human monoclonal antibodies, humanized monoclonal antibodies, and therefore will not generate the kind of antibody immune responses, is a major goal for a very large number of immunotechnologies. So absolutely, the answer to both of your questions is yes and yes. So I hope was that a reasonable answer?
Yes, sir. Thank you so much, sir.
You're welcome. Can we go back to Harsh Pamar?
Thank you, sir. But I actually had the same question.
Oh, okay, great. So, good.
Sir, I do have a question, if you'll let me ask.
Go ahead.
So we know recently in some surgeons in New York practicing in California have conducted the first kidney transplant from a pig to a human.
Pig.
Yes, sir. So, sir, how is that possible? I mean, a kidney of a pig and a kidney of a human are totally different, anatomically very different, physiologically might be different because they both have different type of diets. And then, sir, the receptors on the renal cells are also very different. So, how is it possible? I mean, if we give immunosuppressants to the human, then that will make the human very vulnerable. Then how did that transplant become possible, sir?
Yeah. So the answer to this question is in multiple layers. Everybody does recognize that we're going far away from the immunotechnologies that your workshop is addressing, right? That I don't have a problem with addressing the questions. Just think about all the questions that you're thinking about and how they flow outwards from any consideration of immune system components. That's all. But that said, here's an answer. So in the first place, pig kidneys are actually not dramatically different from human kidneys. Shape, size, function, formation, they are pretty close. The pigs from which these kidneys are being drawn weigh anything between 50 to 100 kilograms, which is what people weigh. And some of us weigh more, some of us weigh less, but kind of sort of thereabouts. So in a sort of organ function fashion, kidneys are in relative terms, the easiest organs to transplant because they have a capsule, they have an ingoing artery, they have an outgoing vein, single artery, single vein that can simply be stitched together, and their output, their urinary output is simply connected to a ureter. So once those three connections are made and they are anchored, they're stapled to the back of your abdominal wall, hey, you've got a kidney. Will it function? Absolutely, yes, it will function. Does it function? Yes, it functions. We've known that it functions for a very long time. So it's not a problem. What's a problem is immunological rejection. And here are, again, layered responses to the immunological rejection. So in the first place, if we transferred, as people did 100 years ago, we transplanted pig kidneys, people transplanted sheep kidneys into pigs, pig kidneys into goats, and all sorts of interspecies combinations. It became very clear very early that human immune responses to pig tissue were massive and extremely rapid. In a matter of hours, the transplanted pig kidneys became inflamed, and you could reproduce this in vitro because pigs have a cell surface glycosylation of protein molecules, both cell surface and secrete products, but a glycosylation that's very different from humans. And it turns out that that is a very common, almost generic immunological target. So it leads to what's commonly called hyperacute rejection. So the pigs from which the latest transplant that you're talking about has been done are not any old ordinary pigs. They are pigs that have been genetically engineered so that their glycosylation patterns, alpha-galactosyl transferases, are not pig transferases but human enzymes. So they show a human glycome in a restricted sense. And therefore, it is thought that the pig kidney that is being transplanted will not evoke hyperacute responses. And in fact, the news item is because the first few days made it obvious that that was indeed correct, that there was no hyperacute rejection. That said, let me come to the second layer of the problem. And the second layer of the problem is that pig tissues will be recognized by human immune system. So will that happen? Yes, it will happen. The question is not whether it will happen or not. Remember the response I gave to Dr. Bashak's question. The issue is not whether there is autoreactivity or not. And in a sense, if I'm a patient and a recipient of a kidney transplant, then me responding to that transplanted kidney is immunologically kind of autoreactivity, right? I mean, we don't want it. So will it happen? Will autoreactivity take place? Yes, it will. The question is, can it be kept within manageable bounds so that the patient can function? And in order to answer that question, we need to think about your problem not in qualitative yes-no terms, but in quantitative terms. So what quantitative terms? Can we allow the patient to live with only mild immunosuppression? Which means that the question becomes, is a pig kidney more or less immunoreactive than a human kidney from an individual who is not HLA identical? Because finding HLA identical donors is practically impossible. So what we are now thinking about is a comparison of two situations. One in which there is a pig kidney coming into me. Second, there is a human kidney from somebody who has a different HLA from mine coming into me. And paradoxically, it turns out that my autoreactive T cells recognizing the pig kidney is actually quantitatively somewhat less than the potency of my T cells recognizing somebody else's HLA molecules, another human's HLA molecules. How and why is that? It'll take, in actual mechanisms, it's going to take a few classes of immunology to describe. But let me give you a little flavor by an analogy. You sit in a classroom and you can look at the faces around you, right? Of your fellow students, and you can distinguish one person from another. You go to a pig farm and you look at a bunch of pigs. Can you distinguish individual pigs from each other?
No, sir.
Think about that. I'm going to stop there. Very provocative.
Sir, I mean, pigs are totally different from us, so they might be able to interact with themselves and they might understand each other, and so it will be easier for them to recognize each other. And as humans, we have a tendency of understanding and recognizing us. So of course, we won't be able to recognize pigs.
In evolutionary terms, our immune systems within each species are shaped for recognition within species HLA. Completely foreign HLA is very poorly recognized in comparison, for the kind of reasons that you're pointing out, that there are consequences to individual recognition within species. There are no consequences to individual recognition between species. I mean, who cares whether it's tiger A or tiger B who's attacking you? Do you see what I'm saying?
Yes, sir.
That's as much as we can do of an immunology class here.
Yes, sir. Anyone else who would like to ask a question? Thank you so much, sir, for your answer and patience.
Thank
With
you.
this, we've come to the end of the inaugural session, and I would like everyone to switch on their camera for a group photograph. Thank you, everyone.
Thank you, everyone. With this, we end the session. And from tomorrow onwards, we are going to start with the lecture session. All the participants are requested to join 10 minutes prior to the starting of the session. Thank you.